Good morning, students. In the previous lectures, we have discussed about the phylum Platyhelminthes, then their classification, and in this lecture, we will talk about the parasitic adaptations in liver fluke. Myself, Balram Sain, Assistant Professor in Zoology, Government Donor College, Bikaner. Parasitic adaptations in liver fluke. Before understanding these, we should know what adaptation really is. Adaptation may be defined as the fitness of an organism to live in its specific habitat or environment. According to Spencer, adaptability is the continuous adjustment of internal and external relations that help an organism so that it can adjust to its specific habitat or environment in which it is living. The parasites are modified morphologically as well as physiologically to live in their particular environment successfully. Some of the parasitic adaptations found in liver fluke are as follows. First is the protective covering. The outer covering of the intestinal parasites become resistant to digestive juices of the host. They develop a thick cuticle. The outer surface of the cuticle is formed of a fibrous protein called keratin. In some other parasites, the outer covering contains scales. Since the cuticle and scales are not dissolved by the digestive enzymes, they protect the parasites. Another is loss of locomotory organs. Locomotion is necessary to search food materials and to run away from the enemies. The parasites live in a medium of food. Inside the host, there are no enemies for the parasite, so there is a loss of organs of locomotion. The muscles associated with the locomotory organs also disappear. Next is the organs for penetration. This is very important. The parasites must penetrate the host body for entering it. So the parasites develop adaptations for penetration. Miracidium has a conical process at the anterior end called apical papilla, which will be more clear by the diagram. This is the Miracidium larva, this is the external structure, this is the internal structure. In Miracidium, it has a conical process at the anterior end called as apical papilla. This is the apical papilla. This is used to penetrate the soft body of the intermediate host, the snail. There are a pair of penetration glands inside the body near the anterior end. Here you can see these are the pair of penetration glands. These glands secrete a histolytic secretion which is used for penetration. Another example for organ of penetration is the free hexanth larva, which has 6 hooks. First, second, third, fourth, fifth, sixth. 6 hooks are present that helps in the penetration so as to enter the body of the host. Next is the sense organs. Progressive parasitism is accompanied by the loss of sense organ. The sense organs are of vital importance to free living animals for they enable them to detect the fluctuations of of different factors in the environment. In the host, the environment is more or less uniform, and so the sense organs are not essential. Hence, they are reduced. The endoparasites are generally provided with the tango receptors. The loss of sense organs is accompanied by the degeneration of the nervous system. Next is alimentation. The digestive tract undergoes simplification, and in extreme cases it is totally absent. The digestive glands are absent. The purpose of digestive gland is to simplify the complex food materials into simpler ones. Since the food is already in digested or semi-digested form, the digestive glands are not necessary for the parasites. Next is anaerobiosis. The intestinal parasites live in an environment completely devoid of oxygen. So parasites are adapted for low metabolic rate which requires minimum amount of oxygen. Moreover, the respiration is of anaerobic type consisting of extracting oxygen from the foodstuffs. In the absence of oxygen, energy is obtained by the fermentation of glycogen, in which by glycolysis glucose is broken down into lactic acid. Next is osmoregulation. The osmotic pressure of the parasite remains the same as that of their host, so that there is no difficulty in maintaining life. Cystodes and trematodes have well developed osmoregulatory system. Flame cells remove excess of water along with waste products. Next is anti-reactions. Most of the intestinal parasites secrete anti-enzymes in order to protect themselves from the digestive enzymes of the host. A dead worm cannot produce this anti-enzyme and is quickly digested by the host. Some of the new medicines aims at nullifying the protective effect of the anti-enzymes, resulting in the digestion of the worms by their hosts. Next is cystogenous glands. The Cercaria larva possess a large number of cystogenous glands in their body. They secrete a cyst around the larva. The cyst protects the larva. That will be more clear by the picture. This is the cysticercus larva. This is the body part and this is the tail region. Here you can see cystogenous gland cells are present, which secretes a cyst around the larva that helps the larva from the effect of the digestive juices. Next is the adhesive organs. The important adaptation met within parasites is the development of organs of attachment. The adhesive organs are in the form of sucker, acetabulum, hooks, spines, adhesive secretions, etc. All these adaptations help the liver fluke in order to survive in the body of the host. Thanks a lot for paying your attention, and in the upcoming lectures we will discuss more about the phylum Platyhelminthes.